Sugar maple populations have been declining throughout eastern North America over the last few decades. The beginning of this dieback lines up with when acid rain was a major concern. Because of this, it's believed that acidic deposition and its after effects are partially to blame for the decline of sugar maples. This study performs cellular analyses on the xylem of 18 sugar maple trees. We expected to see significant changes in length and width of the vessels between interior and exterior wood. This would indicate cellular changes impacting growth, which may be caused by long-term effects of acid rain in soils.
Our study was conducted at Fundy National Park, which is located in southern New Brunswick. This park falls within the Acadian forest, which has boreal-type forest in the north and deciduous-type forest in the south. Fundy National Park is located on the Bay of Fundy, which has significant influence on the climate of the surrounding region.
We were investigating changes in the cellular structure of sugar maples, and these are our research questions. Do sugar maples experiencing radial growth declines, likely due to acid deposition, exhibit changes in cellular structure? We also wanted to know if there were significant differences between our study sites.
We visited Fundy National Park at the end of August and collected samples over a 4-day period. We sampled 3 different hardwood stands throughout the park, which were identified as Northwest Ridgeline, Tipping Lot, and Third Fault Falls. At each site, we sampled 6 sugar maples. At each of our sites, we would go around and identify sugar maple trees, which were hard to come by at times. For each sugar maple that we sampled, we would use increment borers to extract cores. This was quite difficult at times. It was really hard to start the increment borer and get it to bite into the bark. It was really tiring to extract the cores, and even though we were only doing 6 trees at each site, we were all really tired by the end of each day.
Once back in the Mount Allison lab, each core was extracted from its tube, and a razor blade used to take little cuts of the wood from the interior part of the core as well as the exterior. So this would have been closer to the center of the tree, and it would have been older wood as opposed to closer to the bark, which would have been more recent wood. The little pieces of wood that we cut from our cores were then glued to a metal stub using graphite glue. Each stub was divided into 24 sections and then dried and coated in a thin layer of gold and placed inside a scanning electron microscope. 118 micrographs were taken and vessel width and length were measured in order to determine if there was a difference between the wood from the internal part of the tree versus the external part of the tree, so before or after acid deposition, and then statistical analysis was conducted.
The most significant result we found was the change in vessel length from before acid deposition to after. As an example, here are the results from one of our sites, where you can see the length from before acid deposition is around 180 microns, and after acid deposition, it's around 250 microns. You can see there that there is a significant difference in measurements. The significant results that were found in the length between before acid deposition and after were found across all 3 of our sites. There was no significant difference found in the internal measurements across all 3 sites, and there was no significant difference found in the external measurements across all 3 sites. The only significant difference that we found was in the length from before acid deposition and after. We also found a significant difference in vessel width at each site. Here we have one of our sites. So we have the internal wood, which would have been pre-acid deposition, and we can see that the average was about 55 microns. And then here we have external, which would have been post-acid deposition, the wood closer to the bark, and the mean for that was closer to about 65 microns. We also did a comparison for vessel width between sites and found that there was no significant difference for either internal or external. So we can see that these values, the mean was pretty consistent across all sites. So the only significant difference that we found overall was between pre-acid deposition and post-acid deposition.
We initially predicted that the size of xylem vessels would increase from prior to acid deposition compared to after, which was supported. We also predicted that the changes in xylem would be more prominent in some sites than others, which was not the case. This suggests that the significant changes in sugar maple xylem from prior to acid deposition compared to after are consistent throughout the park. Xylem is important for the transportation of water and nutrients within trees. Longer vessels can be detrimental to xylem's conductive abilities, and therefore these can have negative consequences on sugar maple health. These increases in xylem size could also leave them more vulnerable to freeze-thaw events. Overall, these changes could be the mechanism behind the decline in sugar maples throughout the park over the past couple of decades.
The statistically significant results support our hypothesis that the xylem vessel length and width would increase from prior to acid deposition compared to after. While further research is required on environmental factors such as moisture and soil pH, this research suggests that there may be long-term changes caused by widespread acidic deposition. This would leave sugar maples vulnerable to future weather events until soils recover.